situation where they're actually in trans. So having phase can be very important and vital when you have variant adjacency in highly polymorphic regions. There's some solutions to that. We're moving towards graph references. I think this is a good plan. But also we could move towards certainly at least local and in time total de novo assembly to get around some of those issues. And long-read sequencing, I think, is certainly one of the approaches towards that. And what about the accuracy of variant calling? We've been focused in the population discovery era predominantly on single nucleotide variation. But in the clinical world, we've been focused in completely the opposite direction, starting with chromosomal abnormalities and moving backwards towards single nucleotide variation as the less important variation. So it turns out, because our algorithms were developed for population cohort discovery, they're quite good at single nucleotide variation, but actually not that good at the variation much more likely to cause disease. Here, for example, is a simulation showing the difference in sensitivity and specificity, sensitivity shown here, for indel length for haplotype color and unified genotype. Pretty good if the indel is 1 to 3 base pairs, but falling off dramatically as the indel gets longer. Structural variation, we've known also that larger than that is something that we can't easily get to with the algorithms that we have. There are some new algorithms. Here's a couple for indels that have been published in the last couple of years, and I'm sure many in the audience are working on new ones. But again, if you can't see it, you can't measure it. So one of the interesting things, this is from Evan Eichler's paper with the PacBio team published in Nature last year on the hydatidiform mole. One of the important things is that you actually can see into these darker areas of the genome where currently we have no insight, and you find a lot of variation. So from the perspective of a physician sitting in front of a patient trying to get to the answer for that patient, or worrying that I might have the wrong answer for the patient, which is a much bigger concern, then these kind of graphs scare me quite a lot, but also give us some hope, because there are new technologies coming every year, log scale changes in cost and log scale changes in our ability to see variation. And you can see in red and black here, if you're familiar with this, for event size of structural variation that had not been seen before. You can also get to structural variation by doing low coverage whole genome sequencing along with augmented exome sequencing. This is something that the team at Personalis has been looking at, a 1x or 3x or 5x whole genome thrown in, if you like, along with an exome that's augmented for coding coverage. And I think increasingly that's what we're going to need. We're going to need to think of custom assays. To go back to this question about what's the best genome diagnostic today for the patient, I think it's going to be multiple different technologies. And we have an expression where I come from: horses, the right horse for the right course. I say these sometimes and nobody has any idea what I mean. So horses for courses. Let's compare non-inferiority with array CGH, which is the current standard of care for structural variant detection in the pathology lab. And we can find that even this 1x whole genome, thin whole genome sequencing is able to compete with that. So I think we can do a lot better. We can do better than non-inferiority. We can aim for superiority, but we have to get to the former first. So I'd like just to leave you then in the last few minutes with an example of precision medicine in action. This is a particular condition that we focus on in my lab. It's called hypertrophic cardiomyopathy. And you might have heard of hypertrophic cardiomyopathy because it's one of the most common causes of sudden death in young people and athletes. And the reason you can see pretty clearly from this graph on the right-hand side of the left graph shown here is a normal heart with a normal sized chamber and a normal wall thickness. And this is normal ultrastructure. And on the left is a heart from a patient who died from hypertrophic cardiomyopathy with a much thicker wall, a much smaller chamber, and significant fibrosis shown here in blue. Also, the small blood vessels are remodeled. So this is a pretty significant condition. Here's some moving pictures, because I'm a cardiologist. I like to show you people's hearts moving. And you can see here very significant thickness of the wall muscle on this MRI. This is the mitral valve that lets the blood into the main chamber. I don't normally show cardiology things at genetics conferences, so this is kind of fun for me. And if you can see this little valve moving here, you see how it moves up and touches this wall? That's something called outflow tract obstruction. So that means as the blood tries to get out of the heart, it can't because it's blocked by the valve. And that causes significant problems. And here's an aneurysm thinning of the wall of the heart. So this is hypertrophic cardiomyopathy. The therapies we currently have are essentially palliative. They literally involve cutting out a piece of the heart, known as cardiac myectomy. We literally go in through the aortic valve and cut out part of the heart. Or, and this is really true, we inject alcohol to cause a therapeutic heart attack. Believe me, this has actually happened. A therapeutic heart attack to try and infarct part of the heart to take away that outflow tract obstruction. So we had an idea that perhaps a genetic therapy that gets to the root cause might be one way of doing this. It's a dominant disorder, and so there might be some possible benefit from silencing the mutant allele. The problem is we all know that variation is rare, and variation for patients who have conditions are rare. So that means that almost every patient who walks in the door will need a new therapeutic, which isn't going to work so well. However, it turns out that because it's dominant, it's a poison peptide, you could, instead of targeting each of the individual novel variants, you could essentially haplotype the patient and target instead the common variants that are in cis with the novel rare variants. So this new strategy of targeting the common variants would mean that for the major gene that causes this condition, myosin heavy chain, 5 common variants would be enough, so 5 therapeutics would be enough to cover 90% of most populations. So that's good, and we started to work on seeing if it was possible to silence the mutant allele, and it turned out that it was. But then we had the challenge that for each patient we needed to actually haplotype them so that we could target which common variant was required. And given that we haven't yet moved this into real people, we're currently doing it in their stem cells, so iPS stem cells, we had the ability to use the IsoSeq platform to haplotype at the RNA and cDNA level. And so we were able to do that and haplotype the first 8 to 10 patients shown here using this technology, and then do something that we like to call precision gene silencing. So we're actually able to target both the common and the rare variant in order to reduce the expression of the mutant peptide and actually reverse the disease. And I haven't shown here the functional data, but we have data both from the iPS stem cells and from mice with hypertrophic cardiomyopathy that it's possible to deliver this therapy in vivo. So I'd like to leave you with these conclusions. And again, thank you for spending your lunch hour with us. I think precision medicine is really transformative, and it's a really important idea, and it's already here. But for it to be really attained, for it really to attain its full potential, precision medicine also needs to be accurate medicine. The genome is complex. We should never forget that. And short-read approaches have been very successful in many ways, especially in a discovery context. But when we're thinking about the N of 1, when we're thinking about the patient in front of you, where the downside is not that you just don't discover a new variant. The downside is quite literally you make a false call that affects somebody's life in a potential life or death situation. It's a very different use case. And so what is the ideal germline genome today? We need to make sure there's adequate coverage of the genes that we already know cause disease. And so we need to start asking ourselves, can I make a call at every coding position? Because if you can't, that is not superior to our current standard of Sanger-based sequencing. So we probably need some whole genome coverage, which would help with structural variation. And we need solutions, I'm sorry, there's a spelling mistake there, but we need solutions to complex areas of the genome. I think the genome diagnostic that is optimal is going to have to be tailored to the individual complex areas of the genome. I think earlier in this meeting, someone pointed out that there are 100 areas of the genome that are almost as complex as the HLA region. So there are a lot of things that we haven't yet managed to get our arms around. There's some solutions to that, and I think coming to a conference like this, which I always enjoy, and makes me very hopeful for the future of getting to this. If we ask the right questions, I think increasingly we'll find the right answers. Thanks so much for joining us today. I'd be happy to take questions.
So if the audience has any questions. So you brought up a lot of complex issues and problems to bring this whole dream to fruition. What would you prioritize, or what are the first things that we need to do to bring this into the precision medicine?
Yeah, I think that the major thing is to realize that it isn't going to be one size fits all. I think that's the key. We have to think about what the reason is that we're doing an exome or a genome, and think about the specific use case. And I've already alluded to the difference between discovery versus clinical medicine. But I think even within clinical medicine, we have a number of different use cases. Obviously, we're talking about liquid biopsy. A lot of the time, we're talking about germline risk and Mendelian disease risk. So I think that the product, if you like, for that, the optimal genome diagnostic in that area is going to be different, clearly, from tumor sequencing. But I think that the number of ways in which genomics, in all its forms, is impacting medicine already and is going to impact medicine further in the future is really inestimable.
Okay, thank you. Thank you for a very inspiring talk. Before I introduce our next speaker, I just wanted to remind everybody that this is being live streamed on the PacBio website. So you can actually go to the PacBio blog at the PacBio website and see the live stream. In the future, with all these presentations can also be accessed on the PacBio website at the PacBio blog. So now I'd like to introduce our next speaker, Robert Sebra, from the Mount Sinai School of Medicine, the Icahn Medical Center. Bobby, also known as Bobby got his PhD at the University of Colorado, and right from his PhD, he went in and has spent the last 10 years in developing next-generation sequencing. Mount Sinai now has the claims to one of the most complete assemblies by next-generation sequencing of a human genome. He, in a very exciting way, he's also a leader at implementing the Sequel technology. And I'm looking forward at the end here that he's going to show us a little bit of data that he derived from the Sequel instrument. Okay, so thank you, Bobby.
So thanks for the intro, Bill, and the invitation to speak at the Roche PacBio Smart Sequencing session today. It's always great to be at AGBT and share some results with you guys. So specifically, I'm going to talk about some of the great work we've been doing at the Icahn School of Medicine at Mount Sinai around leveraging smart sequencing for better deriving niche assays with diagnostic potential. I say that because most of this is R&D at this point, but we're really driving toward positioning that into precision medicine into the clinic, much like Ewan was driving in his last talk. So before going into any data, I wanted to give you guys an overlay of the technology landscape and why we have this technology at our institute. So this may seem like overkill, but to Ewan's point, when you're working with a diverse array of physicians and healthcare professionals, the diverse arena of disease that you have to tackle is not indeed one size fits all. So you need a variety of toolbox and technologies to address the complex disease that affect the human population. So you can see that we've run everything from many short-read technologies, including Antibody and Illumina, to a strong investment in long-read technologies, including 3RS2s, 2-sequels, and more recently some of the 10x genomics platforms. And we're also trying to fan out toward addressing a lot of single-cell sequencing using the technologies below, because addressing heterogeneity and subclonality is really important for taking an integrated approach. So specifically, what have we been doing with long-read technologies? Our program is pretty diverse. Today I'm just going to focus on the human healthcare standpoint. So you may have noted in 2015 a publication with collaboration with many of you in the audience today, but also Ali Bashir's team, to tackle the NA12878 genome. I'm not going to focus on it today, but piggybacking off of Ewan's comments, it's really important to be able to do a variety of haplotypes to completion so that you do have this arsenal of fully completed human reference material so you can start deriving targeted approaches that are actually precise. So please take a look at that in Nature Methods, and you'll see that using smart sequencing and bio-nanogenomics that we were able to tackle the genome to about an N50 of 30 megabases. And we're pushing forward with many of you in the room for the Genome in a Bottle initiative and others to get more haplotypes featured. So I'm going to talk about the targeted work today, plus a little bit about rapid validation for somatic variation at low frequency. And then, unfortunately, due to time, I won't talk about transition dynamics in infectious disease, but considering how many people I've seen with influenza at AGBT this year, please check out a recent publication with Ellie Gaydon on talking about minor species and how they push the dynamics of transmission. So going into the first talk, part of the talk, much like Ewan showed, there's a big list of disease that we need to tackle that you cannot do so using short reads. In this case, shown in what's showing up on the screen is orange. You can see a list of pseudogenes or genes that have pseudogenes affiliated with them, or CNVs that allow you to basically tackle the disorder and understand the pathogenicity. Shown in red are basically trinucleotide repeat expansions, so everything from poly-Q disorders to an example that I'll show with a hexanucleotide repeat for C9orf72 later. and then in green, polymorphic regions for the pharmacogenomic targets that were discussed in the last talk. So it's really important to understand the allele breakdown across the population to reposition drugs. But all in all, what we've learned from some of the whole genome sequencing aspects, which will be the future, is essentially when you apply long-read technologies that you can interrogate and discover thousands of structural variants beyond 2,500 base pairs that require long reads. The question now is, what do they mean, and how are we going to figure that out across bigger cohorts in time? So we've been tackling a lot of diagnostics at the Institute. In this particular case, please note anything in the pink or red color I'll discuss today, but if you see something in black that you're interested in, catch me later. But it's a diverse array across cancer, immunology, inherited disease, pharmacogenomics, as well as pathogen surveillance, which is really important for differentiating hospital-acquired and community-acquired disease. So I'm going to take you through a half a dozen cases where we've actually started to implement this technology in a more diagnostic landscape. The first example is that of CYP2D6. So much like HLA, which many of you are familiar with, CYP2D6 has many polymorphic features across the population. But what else makes it difficult is the fact that there are many known duplications that are important for repositioning drugs for an individual, but CYP2D6 metabolizes almost a quarter of all the common drugs that we take, so antidepressants, antipsychotics, opiates, and so on. And the diverse array of alleles across the landscape of this gene is just vast. So for you to tackle and correctly phase the variants and understand from star allele to star allele or haplotype to haplotype what's going on, it's really important to be able to do that with long reads. And recently we published this in collaboration with Stuart Scott's lab in clinical genetics, and you can check out the informatics behind that in the publication. But essentially what we've done, as you can see on this slide, is tackle the entire gene in a single product. And in doing that, you can sequence across, phase the mutations, and get a much higher resolution landscape of the gene. How does this perform compared to standard technology? You can see on the left a bunch of Coriell controls, but also a Caucasian and Hispanic sample that we use internally. When you start comparing this both to the reported literature and also to the Luminex and TACMAN assays that are typically used in the clinical setting, you can see that we can correctly call the number of the copy numbers correctly in intron 2 and exon 9, and phase the heterozygosity of these samples quite well. So everything shown in green is correctly called relative to literature. And then in blue and yellow, these are cases where we're starting to see even higher resolution because of the nature of the long-read sequencing. So not only can we differentiate ambiguity from some of the reported literature in some of the blue cases where there was a debate in published reports, but in the yellow case, you can see an additional copy of 2D7 that was actually acquired in the sample that was not previously detected in any of the technologies. So we were really excited to see this and have been starting implementation toward the clinic. So the next example is in the desire to basically phase and differentiate variation between a gene and an affiliated pseudogene. In this case, it's GBA. GBA is affiliated with a disease called Gaucher disease. It's a metabolic disorder, which is a lysosomal storage disorder. It's a spectrum of presentation, which makes it incredibly hard for a physician because the spectrum can go anywhere from no phenotypic presentation to eye movement disorders or even Parkinsonian-like symptoms. But what makes it a genetic challenge that's interesting to tackle with long-read sequencing is in GBA, you can see from the blue track that the GBA gene and the GBA pseudogene, which is only 16 kb downstream, have a lot of commonality other than some novel splicing events, but you also have many mutations that have been reported up to 300 that you have to figure out and differentiate. So to do that, you need to sequence across the entire landscape and then use the flanks to identify whether or not the mutations are occurring in the gene or the pseudogene. And so you can see in this case, we've created a 6.4 kb product that includes really nice flanks. With those flanks, you can guarantee that you're mapping the variants correctly back to the gene or the pseudogene, which is really important if you're creating a diagnostic in this case. And it also helps you understand and string together those mutations across allele to allele. So the third example is a really challenging example for sequencing technologies, which is C9orf72 in ALS and FTD, which is frontotemporal dementia. This is a collaboration with New York Genome Center and Columbia University and Neil Schneider's lab. What we've started to do here is try to tackle the hexanucleotide repeat, which is a G4C2 complex, which, as you can imagine, is pretty much horrible for trying to do amplification methods and/or short-read sequencing. In this particular case, there have even been cases that have shown to have thousands of repeats, but at least anything over 30 repeats is considered to be pathogenic. So between exons 1A and 1B, that repeat exists in the intron space, and it's basically responsible for an accumulation of dipeptide repeats, which leads to an accumulation in the brain of this protein, which leads to more severe disease. So using a sequencing technology like smart sequencing, you can start to tackle this and hopefully move away from the traditional laborious and less resolved technologies like Southern blotting to resolve the repeat structure and come up with a predictive assay in this case. So what you're looking at on the top track and the bottom track are two examples where you have two alleles mixed, where the repeat identity in example one is basically around 50 repeats. And then the second, which is the little peak I have labeled as A and greater than 150, is a very minor allele mixed in there. And you can't see that well on the band, so I have it labeled. But even in cases where you have very rare occurrences or subclonality, you'll be able to sequence through that. And the second case is a little more cookie cutter with repeats between about 40 and 75. But it just shows, if you look on the right, that if you use an assembler, that you can actually error correct the reads and get perfect identity across the G4C2 repeat, which even I was pretty surprised by, considering just fidelity of enzymes. So we were excited to see this and are hoping to push it forward into that community. So I did want to note in this case, since we have started to use SQL over the past couple of months, it's also a good opportunity to show you what some of that data looks like. So if you look on the left, that's the RS2 data. On the right, we actually sequenced the exact same samples using the SQL instrument, and we've been able to prove to ourselves that we're not introducing any weird error phenotype in this case, because we were able to call the very same repeat distributions, and we're excited because now we could start multiplexing and barcoding and putting multiple samples on the same instrument. So it's important to reveal that because you just never know between technologies, and it's good to show that primary analysis is working well. So the next example is actually a rare disease. This is around the subject of oculoauriculovertebral syndrome, or Goldenhar syndrome, and is a collaboration with Bryn Webb, who's an amazing physician at Mount Sinai. Just to give you kind of the motivation behind doing this, in this particular case, it's a congenital craniofacial disorder that just has a huge spectrum, again, of presentations. But what makes it important for us to investigate, even though it is a rare disease, is that it's so detrimental to the individual in terms of all the surgical history that they have to endure across the course of disease. So you can see this individual, their first surgery was on day 7 of life, and then 7 months, and essentially every 5 years after that. So the more information that we can uncover using technologies like smart sequencing to figure out across the population and understand both a diagnostic potential but how the disease works, the better for the community. And so the take-home in this particular project, which we hope to publish later this year, is shown here. It's actually a 1.3 megabase line element that's inserted between 2 line elements on chromosome 14. It's extremely functional in that regard. So if you look at the proband, the biology has introduced the new line element that I blow out and then show on the flanks that you can see a known phenotype of SNPs that identify the correct placement of those reads, but you get this inverted duplication between the control events, which include genes like OTX2, which is a known sort of a driver functionally for Goldenhar syndrome, and Bryn and others have actually taken this into a zebrafish model and shown functionally what this does to the biology, and that'll be presented alongside the data. But to be honest, I don't know of another way you could tackle this type of structural variant other than using long reads. So it's incredibly important to come back and also use orthogonal approaches. So in this particular case, we use jumping libraries to revalidate the breakpoints. So a really kind of strange example in biology that is tackled using long reads that I was excited to work with Ray Young, who's a talented neuro-oncologist at Sinai, is this, which is essentially using inverted PCR products to target damage in double-stranded breaks associated with radiation in glioblastoma. So if you take the traditional therapies, so adjuvant therapy, and this gene RB1, basically upstream of exon 21, there are these known break sites. So in a control case, you actually get 2 break sites, which I'll show on the next slide. But then after therapy, you initiate 4 other break sites, and they're important for the genomic localization of those double-stranded breaks, essentially governs the resistance profile or the emergence of the profile. So you could use that as a way to predict what's going on across the course of therapy. And so looking at the data here, which is another case of showing both RS2 and SQL, so you can do a technology comparison here, but also follow the biology, which is if you look at the top track, after irradiation, those 4 red sites are the sites that are initiated after irradiation. In this case, it's a patient-derived spheroid line that mimics the tumor. And you can see an accumulation of those break sites at the correct positions downstream of the restriction site that was used in this particular case versus the control. And both technologies show that nicely, and you just happen to see, obviously, more reads coming out of the SQL technology, which you would expect. So this is a good use case. And if you really look after therapy, there are also some bins where you're starting to populate what might be potentially novel sites, but we have to do more in the study to figure that out. So the last example I'm going to show in this half dozen before going into validation is for Baraka 1 and 2. So this is actually a project we've been working on for a while now. So in collaboration with Rong Chin and Andrew Uzalov, which is somewhere in the audience, basically we've created a 70 amplicon panel where you can take 3 to 6 kb amplicons and sequence the entirety of Baraka 1 and 2. So this is both a discovery project and one that's meant to potentially be of diagnostic power. So you can imagine if you're using these long targets across Baraka 1 and 2 and you're targeting all 300 kb of the 2 genes, you can discover anything that's going on in the introns and intergenic space. And I'm not claiming that we understand the function of that, but the hope is that we can start compiling that information and use it as a use case across a bigger population of samples to figure that out. But the take home right now is if you go into a database, much like Ewan was pointing out, it's so biased by whole exome sequencing, particularly at low coverage, that you see on the bottom tracks basically an immense amount of data happening across the exons, but not so much in the unknown. And so what happens when you come back, in this particular case, we took 3 cases and we sequenced them, in this case, across, and doing an exon comparison in this case, across the 3 technologies of PacBio, Antorrent, and Illumina, you can see really good concordance between the 3 technologies in the spaces that are covered by all 3 panels, with a little bit of novel information coming out of the PacBio alone. But when you open the floodgates and you basically start looking at all of the novel regions that are starting to be covered by the extensive panel across the entire gene, you then start to see dozens to hundreds of novel mutations, of which some of them are actually annotated in dbSNP, others are not. So we'd have to figure out those functions by doing a bigger cohort, but it's definitely inspiring to figure out what they all mean, because reverting back to the topic of whole genome sequencing, you can see in some of those reports, whether it's from Genome in a Bottle, Evan Eichler's group, or our own publication, that there are thousands and thousands of variants that are popping up in genes that we thought we knew everything about. So I'll leave you with one last example of how we've been using the technology that has really accelerated some of our work, and that is just in our cancer pipelines. So like many of you in the audience, we tend to use a lot of whole exome sequencing, but the risk of using whole exome sequencing alone In heterogeneous samples, obviously the coverage, Ewan kind of broke that down nicely in his talk, but I'm going to show you some real examples where basically coming back and using a hand-curated list of variants that we thought were significant in these cases can overturn some of the conclusions by going in at higher coverage using the CCS reads from smart sequencing. So this example, which you're looking at here, is one of those cases where basically in this case we had tumor normal matching. We had a primary tumor and a MET. In the primary, it was 50% pure, and the MET, it's 10% purity. They're from FFP-derived samples, but you can see the sequencing metrics from the HiSeq, which are decent. They're 100x, 200x, and 300x to try and titrate the coverage. Obviously, that's decent data for whole exome sequencing, but the take-home there was that we had 31 variants of significance that the team was interested in. Andrew Uzalov and others basically annotated those, and you would have left that at, you had 4 germline variants, 27 somatic, and 8 of which were exclusive to one of the 2 tumors, where if you come back and you verify at much higher coverage, you overturn 4 of those conclusions. So 4 of the variants that you thought were only in one of the 2 tumors are suddenly showing up in the MET, and it makes sense because the purity of the MET in this case was so low. So this drives the need of using systems like this to validate, and honestly for us it's been easier to just push these through on smart sequencing than for instance using Sanger. So the hope is that, particularly with SQL coming out, we can basically do extremely high multiplex and start targeting more and more variants in this way after annotation, and then really drive the need so that the precision of the prediction is much, much higher. And I'll leave you with one last result from SQL. In this particular case, one of those cases, I'm just showing the RET gene, which in this particular case is affiliated with medullary thyroid cancer. And that's case A on my chart. It's just anonymized for samples A, B, and C. So in that case, you can see really great concordance between RS2 and SQL. And just to show you what the reads look like from the SQL data, they're not really any errors that are causing any issues with the call there, which is really important in these cases because that's a key driver mutation. You can just look briefly on the table on the right at the concordance between the variants called between the 2 technologies. We really haven't had any issues there, including if you look at the second to last case where the 5% and the 6% mutation are called almost identically. So that's good news, again, to support that there's no additional bias coming into the platform. So we have to keep testing that. So I'll leave you with the fact that, like, hopefully I've shown that, you know, piggybacking off of the first talk, that we've actually started to apply these technologies in some of these functional domains and that we're starting to really resolve more information. Now the goal is to do more human genomes, more haplotypes, better understand fully resolved genomes and tie that back to some of this targeted method until we get to the point where doing long read genomes is completely cost effective in the future of genomics. So with that, I'll take any questions, and thank you so much for your time.
So Bobby will take any questions from the audience, if there's questions. Yes?
Maybe just a technical question to the last slide. So it looked like your SQL reads had a very much higher base quality than the RS2 reads. Is that actually the case, or is that just the CCS correction?
Yeah, so Vince, it's a great question. Basically at this point, I wouldn't necessarily make a global comment based off of the number of runs we've done. In that case, it is the case, and it could be due also to the difference in pipelines. But particularly there, because you're getting more reads, you're definitely getting better CCS quality in that N of 1.
Okay, so that's a between read CCS rather than a proper circular consensus CCS.
These are consensus, like in those calls, because to your question, we're basically creating amplicons that are really short and then going around and around and around to correct. But you could do either way, right? You either need depth or number of passes.
Thank you.
Yeah.
All right, so thank you, Bobby.
Yep.
So our next speaker And thanks, Bobby, for showing what happens now when we start look at the whole gene loci. Our next speaker is going to talk about what happens now when we can look at whole viruses and viruses in phase. So I'd like to introduce Ben Merle from UCSD. He basically got his PhD from the University of Stellenbosch in South Africa. And he's currently funded on a K99, and he works in Davy Smith's lab at UCSD. And they're one of the leaders in studying HIV by exploiting the long reads of the SMART sequencer. So I'll turn this over to Ben.
Thank you very much, Bill. So I'll be telling you about some of the viral sequencing work we've been doing at the Center for AIDS Research at UCSD. And I'll briefly talk about sequencing viruses in general, and then I'll talk about the HIV vaccine problem, which we're hoping to make a contribution to, and a few other directions. So viruses are diverse sometimes, and these are two phylogenies that are equally scaled. And on the left, you have 30 million years of primate lysozyme evolution, and on the right, you have HIV. And this is not all HIV envelopes. This is 4 years of evolution within a single host of HIV. So there's really a lot of diversity here. And you have to sequence that diversity appropriately. And previous strategies were perhaps not quite so appropriate for capturing these viral populations. So up on the top left, you have bulk Sanger approach, where you get this consensus sequence for everything. And then on the top right, you have single genome amplification, where you do serial dilutions and amplify them up separately. And that's really, really great, but you get a handful of reads for an arm and a leg. And then on the bottom left, you have the short read Amplicon next generation sequencing approach, where you sample one small slice of the gene in a lot of depth. And then on the bottom right, you have the shotgun approach, where you get the length in a way and you get the diversity, but you lose any linkage information between sites that are not right next to each other. So you can see where this is going. PacBio is a really natural solution here, where you can sample across the whole gene, and you can sample in substantial depth, and you preserve all your linkage information. And just to convince you that this is not just a nice pictogram, this is what an actual alignment from our envelope sequencing data looks like, and this is at the amino acid level. And I've kept the scroll bar in this screenshot, so you can see there really is a mountain of data here. It's sort of more than you know what to do with. And you can immediately see features of the viral evolution. The nice conserved regions are cleanly sequenced, and then you see all the variable regions where the real evolution is happening. So how do we get data that looks like this? Well, it's really quite straightforward. You go from viral RNA. In this case, I'll be talking about HIV envelope, which is a 2.6 kb gene. You go from viral RNA and you generate your cDNA and you amplify up the region of interest, and then you do the standard PacBio library prep with the SmartBell adapters, and this is all using circular consensus sequences, so this is all analyzed at the single molecule level. And this straightforwardly gives you that really, really nice high quality data. So what are we doing with this data? Well, there's this puzzle in the vaccine literature for HIV, and what you've got is HIV envelope is the only viral surface protein. And it's really diverse, which is not just a challenge for sequencing, it's also a challenge for eliciting antibodies, because most of the time an antibody to envelope only neutralizes the envelope that happened to elicit that antibody in that same donor. So they're very strain-specific neutralization. But some small percentage of the time, and it's sort of disputed exactly how much, donors that are chronically infected with HIV develop antibodies that do have breadth, that do neutralize multiple different kinds of envelopes. And we really have no idea why. And that's what we're trying to make a contribution towards. So what happens is, the initial virus gets in, and you have an antibody response to that, and the virus escapes, and you have an antibody response to that, and so on, and you get this co-evolutionary arms race within the host itself. And the idea is, if you can take the donors that produce these broadly neutralizing antibodies. And you can track them right from early infection all the way through, and you can sample both the antibodies and the envelope side by side. Then maybe we can use those envelopes as vaccine constructs in the right sequence to elicit the same antibody maturation pathway and elicit broadly neutralizing antibodies in uninfected people, and that would constitute a working HIV vaccine. So that's the rational vaccination strategy that most of the sort of antibody HIV vaccine stuff is based on. And of course, what we're going to be talking about most is, PacBio is the perfect natural way to look at these envelope variants in lots of detail. So we've teamed up with the International AIDS Vaccine Initiative, who have one of these longitudinal primary infection cohorts. And they're busy doing a lot of immunological characterization. They identify where these antibodies are binding on the envelope, which is called the epitope, and they're looking at the neutralization breadth in the plasma over time, and they're doing all sorts of other mapping. And we sequence the virus. So there's all this immunological characterization, and we sequence the envelope, and we design custom sort of informatics and analyses that then we feed back into them. So you look through your viral population, and you pick out because these are, it's whole gene sequences, you can pick out genes that you then synthesize and go and, you can functionally validate and see exactly how they're interacting with the antibodies. So we really close the loop quite nicely. So I'll tell you briefly just about two donors that we've studied in detail. So the first donor that we were looking at with PacBio data, they produced an antibody lineage, and you can see a crystal structure of that antibody on the right that targets the apex of the envelope trimer, which is right in the middle at the top. And you can see that the mutations we see changing in the virus, which I'll get to on the next slide, are also functionally validated as escape mutants from particular versions of these antibodies. And this donor is quite exciting because it develops breadth early on in infection, which means that these antibodies don't need to mature that far, which is important. And I think this is only the second apex antibody donor that's been followed since initial infection in the literature. So it's really exciting to see whether you see the same patterns in both of these donors, because anything repeatable is, of course, more exciting. So when we sequence envelope in this donor, you see this is a phylogeny, which is going to give you a nice overview of what's going on here. And what I've done is I've collapsed very similar variants into bubbles and represented them with the bubble size. In early infection, you get homogeneous variants that then sort of break up and expand, and you get this typical ladder-like structure, which indicates within host evolution with selective sweeps to preserve some level of homogeneity. And I should add as an aside, because this is about sequencing, that the little dotted lines around the phylogeny are pointing to clonal sequences. So you can see that those intersperse nicely at the same time point. So we're sampling the same thing here. It's just the PacBio data samples it much more deeply. And of course, the devil's in the details of the sequences. So we drill down to the specific positions we know are important, and the set of amino acids at those positions defines what we call an immunotype there. And what you can do is chart the evolution of these immunotypes over time, which is this bottom panel. And the story that the sequencing tells you here is that you had an initial variant, and that was very rapidly knocked down, probably by an antibody response, but we can't actually pick that up because it's too early in infection. And what follows that is 5 different configurations of different configurations at this epitope region that were all co-circulating at the same time. And that's immediately after that you get the rise of broad neutralization in the plasma. So there are plausible immunological stories you can tell there, like those might all be being recruited into the same germinal center, and that causes the antibodies in there to be optimized for breadth rather than for specific binding. But it's really quite a detailed story, and there's a manuscript in preparation here, so I won't go into too much more detail. The next donor we're looking at produces an antibody response to this glycan patch, and that's important because this is a response you see responses to this patch appear in a large proportion of the cohort. I think it was about 40% of the broad neutralizers had a response to the same site. And this donor had some very interesting viral evolution going on because they were initially infected by 2 founder variants, which is not nicely represented on a phylogeny, but you can kind of see them there. We think what happened is that in the source, there was some superinfection event where they were infected by 2 variants that then recombined, and during the transmission to this donor, 2 of those came across, because they're very similar in some regions and very different in others, and it seems like the only explanation for that. And importantly, they're quite different in the regions where this N332 response happens. So again, it's a similar story that it could be the sort of concurrently circulating diversity that's driving this broad response. But this is actually a very complex case, and you can't really handle this well with phylogenetics. So this is another way of visualizing the population structure here, and you can see this is called multidimensional scaling, and you can see your initial time point. You have these 2 distinct variants, and I'll step through time here. In the next time point, one of them's almost gone. You see only the other one, really. And the time point after that, you get this recombination event that generates a kind of bewildering array of diversity between different segments of these. And it marches off over time, strangely enough, maintaining 2 distinct variants that never outcompete each other and never recombine. And we're trying to figure out what could be going on there from a virology perspective. And so those are the donors we're looking at in detail, and there are more in the pipeline. But the other thing we're going to be doing is looking at larger sections of the cohort all at the same time. So I think we have about 17 of these donors that target this N332 glycan patch, and we're going to be sequencing the envelope in all of those and comparing it to the evolutionary trajectories in the donors that don't target that, and hopefully pick up patterns that are repeatable, that elicit this kind of response. And to do that a little more cheaply, we're multiplexing this, and it turns out that works very well. So this is 4 years of HIV envelope evolution in quite a lot of detail from one donor, but sequenced on a single smart cell run, which is, I think, quite remarkable. And what we did is we sequenced one time point twice, so even at the sort of depth you get with multiplex data, you can see on the A and B panels on the left show that you have very good agreement between the 2 samples. Your between sample distances look exactly like your within sample distances, and you get a really nice correlation for the amino acid frequencies on that bottom left panel. And if you look on the right, that's comparing one time point to the next time point, and you can see it doesn't look the same at all. So it really does sample the population quite nicely, even even when you multiplex 6 time points on a single smart cell. So I'd really like to know how this will look on the sequel. We haven't done that yet because I don't have a sequel, but I'm quite excited. The other thing I'll talk about very briefly is some of the analysis tools we've been building for this, because this data can be quite challenging. And I won't go into the details of the back end, but we've got something called Flea, which is the full-length envelope analysis pipeline, and it'll nicely generalize to other genes from other viruses. But briefly, you take your data off the sequencer, you throw it at the pipeline, and it thinks for a few hours and gives you back a URL. And then in your browser, you can nicely go and interrogate your data. So those immunotype frequency plots, you can interactively generate by clicking on the different positions, and you can add or remove positions and update that, and you can scroll down and see condensed views of your alignments. And you can browse different sections of the gene region. And we also do phylogenetics in the browser now. So you can get nice big overviews on the right, but you can also look in detail at your phylogenies, and you can take the immunotypes and annotate them on the tree, so you can see, do the mutations that change the immunotype pop up lots of times in parallel, or do they pop up once as founders? And you can see all of that nice and conveniently. And the other thing we do is we map a bunch of evolutionary parameters onto an envelope trimer structure, so you can actually animate this over time and see exactly where the action's happening on the protein. So our collaborators get quite excited about that when we give them URLs for their donors. So briefly, some other directions, and this set is extremely new. It came off the machine 3 days ago, but we were shipped a full-length hepatitis C amplicon, so it's 9.2 kilobases. And it was a clean amplicon when it left France, but it was a fragmented amplicon when it arrived in our lab, so we think it was sort of sonication by border customs agent or something like that. And you do see, so this figure on the left is reads mapped to a reference, and you do see in the middle there are full-length reads that span the whole thing, and that's extremely convenient because we can now use those to set up full reference sequences, and we can scaffold all the short reads against those. So a similar story to the genomic analyses, except now you've got a lot of diversity, even within that time point at the full-length level as well. But we really haven't done much on that. And the other project I'll mention briefly is one of the other people in our group is working on sequencing full-length POL. And POL encodes protease, reverse transcriptase, and integrase, which are 3 key regions targeted by antiretrovirals for HIV. And the idea is that patients fail treatments because they develop drug resistance, and phenotypic drug resistance is extremely predictable from the genotype, and HIV genotyping plays a big role in patient management and choosing which drugs to switch people onto. And this would be extremely convenient if you could sequence all of POL, because you'd get all of the mutations at the same time, which is great. And it's also much more phylogenetically informative. So if you want to understand, say, some compartment like the lymph compartment has much lower levels of drug penetration, maybe you can see drug resistance mutations emerging in the lymph and spreading, and having the long reads helps you nail down some of that phylogenetic signal. And there's also the point that not much has been said about linkage between resistance mutations. So it might be the case that you have a minority resistance mutation at this site and another minority resistance mutation at this site, but if they're not on the same strand, then maybe you don't have to worry so much about it, and that could turn out to be clinically important. We don't know yet, and if it is, PacBio could make a unique contribution there. One other thing, because I think I spoke a little faster than I was expecting. My boss in our group is working on sequencing the HIV latent reservoir. So strategies to try and cure HIV need to eradicate the reservoir, and the reservoir is made up of some proviruses that are intact and will produce replicating virus, and some of them that aren't. And it's plausible that you come up with interventions that reduce the repertoire in a way that kills the intact ones but leaves the broken ones. So we're trying to use long-read PacBio sequencing to sequence the repertoire and quantify how much is intact and how much is broken, so that you can actually measure the effect of these interventions. So to wrap up, PacBio seems like a watershed moment for viral sequencing. It's the first time we can see these populations in this much detail. That's quite exciting. So to thank my collaborators at UCSD, the International AIDS Vaccine Initiative, the Scripps Research Institute, and Grenoble in France, and the people at PacBio that have been extremely helpful. And my funding is from the NIAID. Any questions?
Are there any questions for Ben? So one of the questions that I had, though, is what do you see as the other opportunities in virology for sequencing whole viruses immediately? You talked a little bit about hepatitis C.
So I think influenza is a great one, but that might be cornered already. To be honest, most of my experience has been limited to HIV, so I might decline to comment on the rest of it.
And then the other question I had, and you were talking about hepatitis C, do you have any plans to study with the integration type of sites as well, or?
Not yet, as we're only just getting started on that, so.
Okay.
Thank you.
Okay, great. No, thanks, Ben. Okay, so our fourth speaker is Adam Amor. So Adam is from the University of Uppsala in Sweden, and he had his PhD also from Uppsala as well as his postdoc. So as you may know, the University of Uppsala has been a leader in implementing the smart sequencing in Europe. Currently, Adam is the head of the bioinformatics, and he's at the SciLife Labs, which is one of their national genome research centers in Sweden. And as one of the interesting statistics I've learned actually about the SciLife Labs is they literally publish one scientific peer-reviewed paper a day. So today, Adam will speak to us about the clinical smart sequencing and using the smart sequencing from going from single genes to complete genomes. So Adam.
So thank you very much for this nice introduction. And I'm very glad to be here to present some of our work today. And also I hope that I can sort of replace Ulf Julensten, who was supposed to give this talk in a good way. So first, just a few words about the place where I work. I work at the National Genomics Infrastructure in Sweden, and we are a core facility that is open to Swedish researchers, and we provide next-generation sequencing services. Currently, it's about 70 people working at this organization, and we're located both in Stockholm and in Uppsala. We have quite a lot of equipment for doing NGS sequencing, and these are all the instruments that we have at both of these sites. And if you look at this list, we are in fact one of the most well-equipped sites for NGS sequencing in Europe. And we also have quite powerful computer for doing analysis and storage of NGS data. When it comes to smart sequencing, we currently have 2 of the PacBio RS2 systems installed in Uppsala. The first one was installed in 2013, and then we decided to get another one the following year. And so far we've been sequencing more than 3,000 smart cells on these instruments, and they're running pretty much at full capacity all week. We're hoping soon to be able to increase the throughput by using this SQL system that we hope will be delivered during the spring. And what are we doing on these instruments? Well, basically we're sequencing everything that the Swedish researchers want us to do because we're a core facility. And I would say that most of the things that we're sequencing are in fact not human samples. So it's other types of biological samples. And we're also running many different types of applications. But then we have a number of clinical applications that we're also trying to develop at this site. And these are the ones that I will talk about today. And when it comes to these clinical applications, we're doing it on different scales, depending on what type of diseases we're looking at. So we can go all the way from single genes up to larger regions or even to complete human genomes. And when we're looking at these smaller regions of the genome, we need to use some different types of enrichment methods. And currently we're using either long-range PCR, we're also using hybridization-based methods. We've been testing out rolling circle amplification, and also sort of we're developing or trying to develop amplification-free enrichment methods. And of course, when we're doing complete genome sequencing, we are doing just whole genome shotgun sequencing. And today I'm going to talk about some of the applications that we're doing, all the way from single genes up to complete human genomes. And the first example I would like to talk about is a disease called chronic myeloid leukemia. And this disease is caused by a translocation between chromosomes 9 and 22, which gives rise to a fusion protein called BCR-ABL. And this BCR-ABL fusion protein is actually driving this disease. So to treat these patients, you want to use drugs that inhibit the BCR-ABL protein. And in many cases this works just fine, but in some cases the patient can develop resistance to these drugs. And when they are resistant, you want to switch to some other drug that perhaps works better. And this drug resistance is in the form of point mutations in the BCR-ABL fusion transcript. So what people are doing then to try to screen these patients is usually to use Sanger-based methods to amplify the BCR-ABL fusion transcript and to screen for mutation. But that's not really a perfect method for doing it because you need to do a nested PCR, and also the Sanger method is not that sensitive. Perhaps it gives a sensitivity of about 10% or so. So we're hoping to do better using the smart sequencing instead. So we have now developed this workflow for BCR-ABL, where we're starting from total RNA, we create cDNA, and then we amplify a BCR-ABL cDNA amplicon, which is about 1.6 kbs. So we have one primer now sitting in the BCR part of the gene and one part sitting in the ABL part of the gene. From this, we create a PacBio library that we put on the instrument and then do sequencing and look at the mutation results. This is some results from a very early experiment we did, and we get more than 10,000 X coverage normally for the BCR-ABL fusion transcript. At the top here you can see a sample taken at the time of diagnosis of this patient, and these are now the reads that we have, and basically you don't see any evidence for mutations in this case. But this patient has not been treated by any drug yet, so that makes totally sense. But if we look at this patient 6 months later, then you can see this sharp red line here, and this corresponds now to resistance mutations that is found in almost all of these reads. And in fact, in this patient, we also see a weak band here, which corresponds to another mutation that is found in only 4% of the reads. What's interesting about this mutation to the right here was that this one was not detected using Sanger sequencing in this sample, but it was in fact detected a couple of months later, which tells us perhaps that we have more sensitive method for picking up these mutations at an earlier stage. So we did a dilution series to look at this in more detail, and we can clearly see that we find these mutations down to a frequency of at least 1%. And another thing that is very interesting is that we can also look at these mutations in the case where we have several mutations in one patient. We can see if they are compound or if they are sort of sitting in different clones. So to the left here you have a sample from a patient 49 months after diagnosis. We have this mutation at 315, which is present in 91% of the reads. And then we have another mutation that is present in 4% of the reads. But you can see that these are present in different reads also. So this means that we have two different clones in this case. We also have a population of wild-type sequences here, about 4%. And in this patient, 6 months later, two more mutations show up, this one at 276 and this one at 396. And in both of these cases, these new mutations are occurring on the same molecule as the 315. So we don't know exactly what this means, I mean from a clinical point of view, but I'm sure that it's important and we haven't really seen this type of information before. I can mention that we're also seeing, in fact, lots of splice variation going on in this gene, and there's a poster about that if you want to know more about those things. So we tried to develop this method into a more clinical workflow, and we have now an analysis method for screening these mutations. We started from CCS reads and then apply a new tool that we call Kava, and that's a very simple tool that just counts the occurrences of wild type and mutation sequences for a known list of resistant mutations. So in this case we have a wild type sequence up here, and we count the number of occurrences of this sequence in our reads, and we can also count the number of occurrences when this base here has been substituted to a T in this case. And for each mutation we can then classify them either to be, if we have lower than 1000x coverage, then we call this unresolved. So we really want to see high coverage here. And if we have higher than 1000x coverage and at least 0.5% mutation frequency, both on the positive and reverse strand, then we call this a positive mutation. And otherwise we classify this as a negative mutation. We also have a web system for sharing this information back to clinicians. So we can upload these results directly into the system. Oops. Let's see, I can go back. And we have now here as rows, we have different samples that we have been running, and as columns we have different mutations. And we can go in and look at one particular sample, and then we get more detailed information here. In this case we have 2 positive mutations, and in the case where we have more than 1 mutation, we can also get information directly about the clonality of these mutations. So basically here we have counted that this 315 mutation and the 359 mutation, I mean, we look at each read and see if they are wild type or mutation, and then we can compile tables like this. In this case, these 2 mutations then are seen to occur on different alleles. And we've been implementing this now into real clinical routine. So we have a routine service with sequencing up to 4 samples a week. We've been sequencing more than 100 patient samples so far, and in all the samples that we've been running, we have picked up all the mutations that are also found by Sanger sequencing. And what we're working with right now is to lower this threshold to go down in mutation frequency detection to 0.1%, or perhaps even lower, and we think that it would be possible using the CCS2 algorithm. And now that we have this workflow up and running, we're thinking about other applications where we can do the exact same thing. And the next project we're starting is to do this in hepatitis C to monitor drug resistance in the HCV genome. And we're going to do this as a clinical routine screening of the NS5A gene. And we already have a proof of principle that this type of sequencing works. Another thing that we will look into is the TP53 gene in different types of cancer, because it's also a gene where it's important to look at low frequency mutations that sometimes can show up as negative in Sanger sequencing, and perhaps also the clonal composition of these mutations. And we're not only doing amplification-based sequencing using PCR, but we're also trying to use other methods. So we've been developing also a hybridization protocol based on the Agilent probes in combination with PacBio sequencing to capture longer regions. This is one example of target enrichment that we have been running using this approach. And here now we have designed probes over a region that is about 100 kbs. And we have probes up here, and here you can see the reads that we get from this type of target enrichment. So you can see that we can get quite continuous sequence over long regions using this approach. And there are some interesting features of these types of capture. One thing that we think is interesting is that sometimes when we have gaps in the design, we don't have any probes, we can still span across these gaps using the long reads, because of course these are long reads and they can stretch into these regions that are also repetitive and so on. Another thing that we noticed was that in some cases we can get off-target capture. This is showing one gene that is called MICB, and this one doesn't have any probes designed against it. But we still get quite high coverage here. And the reason that we get high coverage is that we have another gene in this design called MICA. So this is the plot to the right here. And this MICA gene is very similar to MICB. So what's happening is that the probes designed for MICA is pulling down regions for the MICB gene, and we can align these reads back now uniquely either to MICA or MICB. But the problem with short reads is that this would have been a mix of the reads. I would also like to mention one research project that we're working on in a region that we think are quite interesting, called the FADS region. This is a region that has been under selection during human evolution, and it also regulates the production of omega-3 and omega-6 fatty acids. So in this region there are genes that are active in the conversion step of precursor fatty acids found in vegetable oils to sort of longer chain fatty acids that are found in fish, for example. And what we've seen is that there are two main haplotypes present in the human population. So there's one ancestral haplotype, and then there's one derived haplotype, which can do this conversion more efficiently. And also this region has been associated to many different traits, like lipid levels in the blood, and also diseases like cardiovascular disease and inflammatory diseases. And you can see also that there is a geographic distribution of these alleles across the world. And our hypothesis is that this D haplotype appeared sometime perhaps 100,000 years ago, and then when you have this D haplotype you can make more efficient conversion, but nowadays this conversion might be in fact a disadvantage and lead to some diseases. And what we have now done is to identify actually the functional variant in this project, which is located in a repeat in the FADS1 gene here. But what we wanted to do is also to see, okay, is this functional variant on the ancestral or is it linked to the derived haplotype? And we did this using long read sequencing because short reads couldn't really span across this region. And what we have been able to do now is to link this variant to variants on the D or A haplotypes. And what we now know is that if you have an A at this position, then you also carry the D haplotype, and this increases the FAD1 activity. But if you have a G insertion here, then you are the ancestral haplotype, and you have lower FAD1 activity. So we think that this is quite important, and perhaps we could even include this fast genotyping in dietary recommendations. One thing that I can just quickly mention is that we're doing rolling circle amplification for certain types of enrichment, and we have been doing it for enrichment of HPV, which is a circular DNA virus that is causing cervical cancer. And what we can get from this type of experiment is accurate genotyping of HPV infections present in a sample, and also we can in fact read the entire HPV genome in a single read, because the HPV genome is about 8 kbs in size. Okay, the final topic of my talk is what we're doing in terms of whole human genome sequencing. So in Sweden, there's an initiative to sequence whole human genomes, as in many other places in the world. So we'll be sequencing about 10,000 individuals a year. And as a part of this project, we're also generating a genetic variant database to sort of capture the genetics of the Swedish population. And in this project, we want to first identify a cohort that reflects the genetic structure of Sweden, and then generate whole genome sequence data, both using short and long reads, and then to make this information available to the research community and also to clinical labs. And the cohort that we decided to use was the Swedish twin registry. And the samples in this cohort are based on twinning, which is quite evenly distributed across the country. And also these individuals are not more healthy or not more sick compared to the general population. And in this cohort, we already have 10,000 individuals that have been analyzed using SNP arrays. And the idea will be then from these 10,000 individuals to identify 1,000 individuals to put into this project. Here you can see sort of the PCA plot showing the distribution of the genetic variation in Sweden compared to other countries in Europe. And in fact, we have quite a lot of variation going on here in Sweden. And if we zoom in just to the Swedish samples, you can see that we have very much variation going on in northern Sweden, whereas other parts of Sweden is clustered more tightly. And what we will be doing now in this cohort is first to generate 30x Illumina data for all these 1,000 individuals. And then we will also select a few individuals, about 3, to do PacBio de novo sequencing. And then as a third step, we will also thinking about sequencing the HLA gene and other clinically relevant loci to get accurate phasing for all these individuals. This slide just shows some data for the first Swedish genome that we sequenced. So this was a 20 kb library sequenced on 157 SMART cells. We were able to generate 45x coverage of the human genome, assembled this into about 8,000 contigs and 2.8 gigabases of data. So I would say that this is quite good results. And now that we have this data already and also are generating the Illumina data, we can start to ask some questions. And the first thing that we will look at is which complex regions can be resolved using this PacBio data. And also we can start to look into what is the value of creating this type of local reference, because we will align all the Illumina data both to our reference that we created and also to the available reference in the database. And so we just started to do this type of whole human genome sequencing, and we think that it's something that will be important in the future. And the simple reason is that we get more high quality genome sequences in this way. We can resolve repetitive regions, annotate unknown genomic regions, get haplotype phasing, and so on. So I have to agree with this quote from Jim Lupsky that the ultimate goal of this will be to do de novo clinical sequencing. And this is a slide just showing the people who are actually doing the sequences. I can mention Ulf Gyllensten, who is the platform director here. And thank all of you for listening.
Thank you, Adam. I don't know if there's any questions from the audience for Adam. So one of the questions that I had, Adam, is you talked about doing the high number of, say, Swedish genomes. But I was wondering how, that's part of how you might handle it, but how are you going to handle your, say, variants of unknown significance in your reporting, say, for the BCR-ABL? What are your plans?
Yeah, so right now we're actually, what we do have is that we're screening the relevant loci. I mean, that's what goes into the report. But at the same time, we're doing an alignment just to look in an unbiased way and also see if we can find other variants. But these ones are sort of, I mean, they're in the list, but they're not sort of reported. They're more just used to communicate back to the clinicians and then perhaps to get a more idea if they are relevant or not.
Okay, thank you. So before we conclude, we were going to invite Jonas Korlak, who's the Chief Scientific Officer of PacBio, to basically conclude our workshop over the next few minutes.
Great. Thank you, Bill, and thank you so much for coming on behalf of PacBio. We really appreciate that you took the time for lunch. And I would like to just spend 3 minutes on some concluding remarks, in that I think in the theme of this workshop of uncovering and revealing some of the things that are still hidden from us and need to be discovered, and some of the things that we already know about but maybe are more difficult to get at with the short-read technologies. I think all the 4 speakers really powerfully highlighted that there's still a lot of work to do in the research space, and then moving towards, as a foundation, to then move towards the clinic. And I think this was also perhaps nicely highlighted in this editorial a few months ago in Nature Genetics entitled Whole Genome ? Where the editors of the journal said that it's time to stop thinking that merely more DNA sequencing, by that they meant short-read DNA sequencing, will give us the variants that determine human traits. And they noted, of course, that this is because our genomes come in combinations of variation that have many different spatial scales, not just single nucleotide variants, and highlighted that one of the potential solutions to get more information, more comprehensive information about genomes, is through smart sequencing. And so in this context, I think it's worth highlighting that there are many different things that you might want to know about your genome, in addition to single nucleotide variation that I've put on the top. Structure variation was mentioned several times, and we know that 75% of all the variant bases that make you different from me are not contained in SNPs and are thus not captured with the so-called $1,000 genome, which is focused on SNPs. Phasing the haplotypes, difficult regions that you might have to gap fill, methylation, and then, like Jim Lupsky said, going towards de novo assembly. And so in this context, Illumina is quite good in measuring single nucleotide variation, but by itself it's perhaps not so good in capturing some of these other characteristics that you might know about the genome. And so it's possible perhaps to add some upfront sample preparation technologies or some other tricks to get at these kinds of phenomena more or less well. But we believe that one of the strengths of smart sequencing is the ability to get at this type of information all in a single experiment with a homogeneous and a one data type structure. And so that's, of course, because of the inherent fundamental performance characteristics of smart sequencing that give you, at the same time, very long reads, but long reads is not enough. Very important is consensus accuracy, the absence of systematic error. Very important is the absence of GC bias, being able to sequence highly AT-rich DNA, highly GC-rich DNA, low complexity sequence that really is the foundation to sequencing actually the whole genome, and not just the genome perhaps that's in a GC% range that's accessible with other technology, and then looking at the epigenetic modification. So in this context, we're very pleased to now introduce the SQL sequencing system, which provides higher throughput based on the same performance characteristics that you're already used to for the RS2. And so a couple of screenshots on the bottom. Tomorrow afternoon in the conference session, I will talk more about the particular data structures, and will show more example data in addition to what Bobby already showed. So one last point that I want to make is that fundamentally the SQL system differs from the RS2 in one aspect. in the performance and the underlying technology, it's exactly the same. Where it's different is that the system is fundamentally scalable. So on the RS2, we kind of hit the limit on how much multiplex you can achieve. With SQL, that is no longer the case. So we believe that over time we can scale the system to provide even more multiplex and drive the throughput as we have in the past through sequencing, chemistry updates, through software, through sample prep, and read length improvements, now also on the instrument, on the chip side, with improvements and increases on the multiplex of ZMWs. So with that, I'd like to leave you and hope that you can, that we'll have a lot of discussions in the next coming days. As I mentioned, I have the talk in the afternoon tomorrow. We also have a party tonight at 9:30 in the Fairway Lawn, just behind the hotel across the bridge. We hope to see you all there, and we all look forward to the discussions over the next couple of days. Thank you very much for coming, and have a great time at the rest of the conference. Thank you.